Hello, everybody. Today we are speaking about characterizing neurons and brain organoids. This is breakthrough science that we are utilizing to find a cure for FOXG1 syndrome. So let's get into it. So number one, why is this so important? What we need to do is actually go into our children's brains, because that is the organ that's affected by this syndrome, and understand what is going on within brain cells and then test therapies. But we can't exactly cut open our children's brains and do these types of experiments. Luckily, there is another way. So many of you have submitted a skin sample to our FOXG1 Biobank. We thank you for that. That biobank is controlled and governed by the Coriell Institute and generously funded by the National Institute of General Medical Sciences. And what Coriell does is they take those skin samples and they take out the fibroblasts from those skin samples, and they grow those fibroblasts and store them. And because of that, we can actually then pay scientists, which we're doing in our research projects, to take those skin fibroblasts and then reprogram them back to their embryonic state, which is basically going into the stem. And that's why it's called a stem cell. So you're literally, you imagine a plant, we're going back to the stem. Once you go back to the stem of a cell, you can build any cell you want: brain cells, kidney cells, heart cells. So in our case for FOXG1 syndrome, we can create something like a 2D brain neuron. And you see a picture there in a petri dish. And that is literally a brain neuron in a little dish. And you can see the little synapses and neuronal activity that we can then test on. But because we're a FOXG1 Research Foundation and totally awesome, we're taking this one step further. And we're taking those 2D induced pluripotent stem cells and turning them into mini brains. And so you could see on this picture that those are literally little brains that are on a dish. So instead of hacking our children's heads open and going into their brains, we've literally created their brains in a dish. And this is really revolutionary technology that has been spearheaded by one of our key scientists, Dr. Allison Motri at UC San Diego. Now, many of you may ask, all right, great, we have these, let's just start testing drugs, let's get on with it, And there's a reason why we can't do that. There's a step before that is very critical, and that is called characterization. Another word that you may hear people say is phenotyping. And what both of these words basically mean is documenting how the FOXG1 organoid is different from a normal, or other words for that are control or wild type. So what we would want to do is document things like, okay, how is the brain structure of this 3D FOXG1 organoid different than a child that does not have a mutation in their FOXG1 brain? What are the different cell types that have emerged? Is there abnormal cell death in brains that are affected by FOXG1 syndrome? What about the proliferation of cells, which basically means how do cells grow and divide? We know FOXG1 syndrome really affects cell proliferation, so we want to document that. What about the formation of synapses between neurons, which basically just means how well do cells communicate within one another? And then overall neuronal activity, which is just like the electrical activity in our brain that helps cognition and helps us think. What are those changes in a child that has FOXG1 syndrome versus a child that doesn't? And only when we can document those changes in a really precise manner are we ready then to go ahead and test drugs. So once we have that documented or characterized, we can then test small molecule drugs, gene therapies, etc., on both our 2D neurons as well as our 3D mini brain organoids. And then we can see, all right, has something changed? Has neuronal activity improved? Has are the synapses more connected with this drug? Is cell proliferation happening at a faster rate because of this drug? And those are the things that we really want to do, and why it takes time, because we have to grow the cells, grow the organoids, test them, characterize them, etc. But this is the year that we are going to see results from these studies, and we're very excited. If you want to learn more, please go visit our FOXG1 Research Projects page, where we talk about this project and others in great detail. Thank you for joining me on this exciting scientific presentation.